लास्ट क्लास हाम्रो कहाँसम्म सकिएको थाहा छ कसैलाई? एन्जाइम्स पढ्दै थियौं। What was your last class? We have already completed enzyme specificity. The mechanism of enzymic action is also done, and cofactors, coenzymes is also completed. So these are different cofactors and coenzymes that are involved in biochemical reaction, organic and inorganic cofactors. We were on the structure and function of cofactors and coenzymes. So in this also we have completed some of the cofactors, their structure and function. So first is thiamine pyrophosphate. So I will not going to tell about these things. So thiamine pyrophosphate is a structure of TPP, which is derived from vitamin thiamine, which is a vitamin B1. Attendance. Start recording. Then FAD, FMN, this is also completed. So this is the cofactor which is involved in oxidation reduction reaction, and this is the structure. This one is also completed. NAD, NADP, which is derived from vitamin B3 niacin, and they are also involved in oxidoreductase reaction. The substrate is either oxidized or reduced, and these cofactors, they undergo reversible oxidation reduction. They act as a temporary electron or proton carrier. So coenzyme A, this one is also completed. Is derived from the vitamin B5 pantothenic acid. Their main function is metabolic integration in the form of coenzyme A, that is they help in the activation of substrate, or they form a very important molecule which is called acyl carrier protein. So acyl actually means the activated fatty acid. So they will link up and they will hold the substrate in position in the active side of molecule or whole substrate is formed. This acyl carrier protein does not allow the substrate to escape the active site of an enzyme. Now we were actually on pyridoxal phosphate. Now we will start from here. So pyridoxal phosphate, this one is a pyridoxine, is a parent compound, and this is the activated form, phosphorylated form of vitamin B5. So B6. Pyridoxal phosphate is aldehyde form. Although there are other isomers of pyridoxine, pyridoxal, pyridoxamine. So pyridoxine or the aldehyde form is the active form. Involved in amino acid metabolism. The main role of pyridoxine is the amino acid metabolism. And the biochemical reaction it is involved is decarboxylation. Mainly, whenever the carboxylase group has to be removed from the substrate, then there is a need of pyridoxal phosphate. It acts as a carrier of carboxyl group. So in addition to decarboxylation, these are the enzymes: glutamate decarboxylase, dopa decarboxylase. It is also involved in transamination, that is transfer of amino group from one amino acid to another keto acid. Deamination, example dehydratase, is required to release amino group from amino compound. Transsulfuration, transfer of sulfur group, and condensation. For example, during the heme synthesis, ALS synthesis is an example. And also an important cofactor of glycogen phosphorylase. So large amount of pyridoxal phosphate is stored in the muscle as a cofactor of glycogen phosphorylase. Decarboxylation, transamination, deamination, transsulfuration, condensation, and glycogen phosphorylase. So remember these 6 mechanisms in which pyridoxal phosphate or vitamin B6. So to remember, it is a vitamin B6. So it is involved in 6 different types of biochemical reaction. And this one is the structure. Another is biocytin. This vitamin resembles a capsule. It has imidazole ring and a thiopine ring, and there is a valeric acid which is actually bonded with the enzyme. The side chain of biotin binds with the amino group of lysine residue of carboxylase enzyme, and this is the site where carbon dioxide will bind. So derived from vitamin B7, also called biotin, and the enzyme linked biotin is called biocytin, and it is required for the carboxylation reaction. It means that whenever a carboxyl group or CO is to be attached to the substrate, then there is a need of biotin. Without biotin, carboxylation does not occur. And most significantly, there are 4 carboxylases that requires biotin. These are most important carboxylase, pyruvate carboxylase, protoxylase, and beta-methylcrotonyl coenzyme carboxylase. So all these 4 carboxylase requires biotin for the reaction. In the absence of biotin, the carboxylation does not occur. Another is tetrahydrofolate. It is derived from the folic acid, also called vitamin B7, and it is a carrier of 1 carbon fragment. So whenever there is need of transfer of 1 carbon, so it may be in the form of formyl, methyl, methinyl, methylene, or formimino. These are the different 1 carbon units. So if there is need of transfer of these kinds of 1 carbon unit, then we need tetrahydrofolate. And also involved in amino acid and nucleotide metabolism, like the major reactions are conversion of serine to glycine or vice versa, synthesis of thymidylate, which is required for DNA synthesis, catabolism of histidine, synthesis of purine, synthesis of methionine from homocysteine. These are important reactions in which tetrahydrofolate is required, and in all these reactions this 1 carbon unit has to be mobilized, and for that we need tetrahydrofolate. In this diagram, this is a formyl group, formimino group, methyl, methinyl, methylene, and methyl group, and all these 5 units contain 1 carbon in different form. So if a reaction requires the transfer of this 1 carbon unit, then there is requirement of tetrahydrofolate, which is an active form of folic acid. This is the structure of tetrahydrofolic acid or tetrahydrofolate. It's called tetrahydrofolate because there are 4 protons or 4 hydrogens attached at 4 different structures, nucleus. So like in the 6, 7, 8. So tetra means 4, hydro means hydrogen, and folic acid. And this is a structure of complete folic acid. So that's why it is called tetrahydrofolate. And the complete folic acid is given in this structure. This is a pteridine nucleus. This one is para-aminobenzoic acid, and this one is a glutamate. And overall this forms the stearic acid, which is also called the folic acid. This diagram shows the involvement of tetrahydrofolate in different biochemical reactions, which I have just told you in the previous slide. For example, in this diagram there is involvement in folate, different tetrahydrofolate that are in different 1 carbon units. For example, during the conversion of homocysteine to methionine, there is requirement of tetrahydrofolate. During the synthesis of thymidine phosphate, which is required for purine synthesis, there is involvement of tetrahydrofolate. You can see here 5,10 methyl tetrahydrofolate is involved here, and again the active form tetrahydrofolate is regenerated. Again, for the formation of purines, formyl tetrahydrofolate is required. So in this way, during the conversion of serine to glycine, again there is tetrahydrofolate is involved. Serine to glycine, formation of methionine, formation of thymidine, formation of purine. So in all these reactions, tetrahydrofolate is required. So you will study in more detail when you deal with individual metabolic process, how this tetrahydrofolate participates in synthesis of these kinds of substances. But for now, just remember, you don't need this diagram, okay? You just have to remember this one, the main biochemical reaction. So serine glycine, synthesis of thymidylate, histidine, purine, methionine. So this one is important. And this one structure is also important because you must know the basic structure of all biomolecules. So then you can go on with other things. So again, this is the structure. This is the structure of methyl tetrahydrofolate, and this one is the tetrahydrofolate which you just saw in previous slide. Another is methyl cobalamin or deoxyadenosine cobalamin. So this one is the structure of cobalamin. You don't need this structure. It is very complex structure. You won't be asked to draw the structure. This is a methyl cobalamin. This is a choline ring structure that contains cobalt. So it is a cobalt containing vitamin, we can say. And this one, the whole structure, this one is the dimethyl benzidine, DMB. So if and this cobalt has 6 coordinate bonds, 1 2 3 4 5 6. The fifth coordinate bond is attached with the dimethyl benzidine, whereas the 4 bonds, they are coordinated with the nitrogen group of this choline ring. This one is the choline ring, and the 6th coordinate bond is coordinated with the methyl carbon to form the methyl cobalamin. Whereas if this 6th coordinate bond is linked with the deoxyadenosine, deoxyadenosine is linkage between the cobalt and the another R group. If it is methyl, it is called methyl cobalamin. If it is deoxyadenosine, this is the structure of adenosine, it is called 5-deoxyadenosine cobalamin. Both of these forms of vitamin B12 are the active form of cobalamin. So cobalamin is involved in only 2 reactions. However, these 2 reactions are very important. One is methionine synthesis, that is conversion of homocysteine to methionine requires vitamin B12, and another is methyl malonyl coenzyme mutase. In this reaction, there is a formation of succinyl coenzyme, which is derived from the metabolism of oats and fatty acid, certain aliphatic amino acids, and certain pyrimidines. And this succinyl coenzyme, which is formed from this odd number of carbon, will be will then enter to the Krebs cycle and will be metabolized through it. And these are 2 important reactions where it is involved. Although there are only 2 reactions, of cobalamin is very important. There is adenosine triphosphate, abbreviated as ATP. This is a non-binding cofactor. This is the structure of ATP. This one is adenine, which is a purine. This one is the ribose sugar, a usual ribose. And these are the 3 phosphate groups. Adenosine triphosphate, also called ATP. If there are only 2 phosphate groups, it is called ADP. And if there is only 1 phosphate group, it is called AMP, or adenosine monophosphate. So all these 3 can exist. So ATP can break down to ADP or either to AMP. And when these phosphate groups, they break apart, then they will release large amount of energy that can be used to drive any kind of energy requiring reactions, also called the endergonic reaction. So as you know that ATP is a universal energy currency, it carries with it large amount of energy, which upon hydrolysis it is released, and that energy is used to catalyze the condensation reaction, usually when the bonds has to be formed. Mainly involved in the kinase synthetase reaction. So whenever during metabolism, whenever you see kinase enzymes or synthetase enzymes, then you have to remember that there is requirement of energy, and when there is requirement of energy, you have to remember there is need of ATP. By the breakdown of ATP, it releases large amount of energy that is used for the condensation type of reaction. Another is lipomide. This is derived from lipoic acid, which is a sulfur containing a fatty acid. This is a lipoic acid. This one is the structure of lipoic acid, and it exists in both oxidized and reduced form. So oxidized form is the lipoic acid, whereas the reduced form is called dihydrolipoic acid, because you can see here two sulfur. They are linked up in the lipoic acid, but as it gets reduced, they will separate and exist as individual sulfhydryl groups, and it's called dihydrolipoic acid. And this, similar to biotin, lipoic acid is also linked with the enzyme, and in its linked form it is called lipoyl lysine. Okay, this is a lipoic acid, lipomide, and this is the lysine side chain of an enzyme, and there is a amide linkage between lipoic acid and lysine residue of enzyme. And its role involved in the oxidative decarboxylation in enzyme like pyruvate dehydrogenase and alpha ketobutyrate dehydrogenase. It means that whenever the substrate has to be oxidized and at the same time decarboxylated, then there is need of lipoic acid or lipomide, and it acts as a swinging arm and carries the substrate between active site in the multi-enzyme complex. So multi-enzyme complex means a single enzyme having multiple catalytic activities. There is the same enzyme and it has multiple roles. Usually what happens, a single enzyme performs single function. It has a single active site, but in multi-enzyme complex there is a single enzyme but it has multiple active site, and the substrate has to be channelized from one active site to another active site. And the role of lipoyl lysine is to simply hold the substrate in position and transfer the substrate between these active sites. So until and unless the whole substrate has been transformed to product, it holds the substrate in position. It does not allow the substrate to escape the active site and release incomplete. So this is the function of lipomide. Okay, then this is all about the coenzymes. If we study different types of coenzymes, cofactors, their structure and function. So I hope you have understood this. If you have any confusion, you can ask me. Okay, now let's discuss about enzyme inhibition. So inhibition means these are the inhibitors which decrease the catalytic activity of enzyme. They can be organic or inorganic. Like organic means carbon based, and inorganic means normally metals, minerals, ions, vitamins, etc. Or they can be cellular metabolites which are produced during normal metabolism that will affect the activity of enzyme, or can be external agent like therapeutic drugs or toxins. They all are, and enzyme targeted mechanisms are more widespread mechanisms in the biochemical reaction. Three categories of inhibition: reversible, irreversible and allosteric. Now let's first see about reversible inhibition and its type. As the name implies, the bond between inhibitor and enzyme is usually non-covalent, so that it can be easily broken. And inhibition can be reversed, that is the inhibitor can be removed out from the enzyme because it is non-covalently linked with the enzyme, so that it can be easily removed out. So that's why it is called reversible inhibition. Now there are different types of reversible inhibition. The first is competitive reversible inhibition. Here the enzyme and substrate are structural analogs, also called the antimetabolites. So since they are structurally similar, structurally related, they compete for the same active site. You can see in this diagram, this one is an enzyme and this is an active site. You know what active site means, which binds the substrate and transforms it to product. And this one is a substrate, this one is an inhibitor. Now you can see here the structure between substrate and inhibitor, they are almost similar. However, they are not completely the same molecule. Their structure is almost similar, not same but similar. So the active site cannot discriminate between the substrate and inhibitor. It thinks that both of them are its substrate because they are structurally related, structurally similar. And both substrate and inhibitor, they compete with each other for the same active site. And it depends either if the concentration of substrate is much higher, then substrate will bind with its site in preference to the inhibitor. On the contrary, if the concentration of inhibitor is much higher than substrate, then inhibitor will win the race and it will bind with the active site. When substrate binds the active site, it will be converted to product. But if inhibitor binds with the active site, this will inhibit the enzyme. But fortunately, the inhibition can be reversed. That is, you can see in this diagram, the active site is bonded with the inhibitor and the enzyme inhibitor complex is formed. But here, if we increase the concentration of substrate, the substrate will easily displace this inhibitor and itself get inserted inside the active site. So now the inhibition can be reversed. So that is the competitive reversible inhibition. The inhibitor and substrate are structurally related. There is a competition between inhibitor and substrate to bind with the same active site. And the inhibition can be reversed by increasing the concentration of substrate. That is, the substrate will displace the inhibitor if the concentration of substrate is much higher than the inhibitor. So in this case, the reaction velocity is usually not affected. Although the enzyme substrate affinity decreases because of the presence of inhibitor, but the working efficiency of enzyme is not affected because as soon as it binds the substrate, it will effectively transform it into product. So this one is the competitive reversible inhibition, and in this table you can see examples that are based on competitive and reversible enzyme inhibition. For example, succinate dehydrogenase, the usual substrate is these two are structurally related. So if the succinate dehydrogenase binds malonate, then it will convert it into product. Another is HMG coenzyme reductase, that is involved in the cholesterol biosynthesis. Substrate is HMG coenzyme. The inhibitor or the structural analogs are atorvastatin, lovastatin. These are the statin drugs which are used to reduce the level of cholesterol in blood. So enzyme. So this converts angiotensinogen to angiotensin, which has role in maintenance of blood pressure and vascular tone. And these are the inhibitors: captopril, enalapril. These are the antihypertensive drugs which are used to competitively inhibit angiotensin converting enzyme. Another is vitamin K epoxide reductase. This enzyme is required to generate the active form of vitamin K. So oxidized vitamin K is the usual substrate. But there is one drug, dicumaral, which is a therapeutic drug, which is an anticoagulant. So in the presence of dicumaral, it binds with the vitamin K epoxide reductase and prevent binding of vitamin K with the enzyme, so that the active form of vitamin K is not regenerated. So it is used as an anticoagulant because vitamin K is required to activate the blood clotting factors. In the absence of so the clotting process does not occur. So that we can say dicumarol is an anticoagulant. And another is steroid synthesis in bacteria. This is required for the synthesis of folic acid, and para-aminobenzoic acid is a usual substrate. But in the presence of inhibitors like sulfonamides, which are widely used as antibiotic during bacterial infection, they will competitively inhibit synthesize folic acid. In the absence of folic acid, there is no synthesis of nucleic acid. In the absence of nucleic acid, bacteria does not die. So this is an example of competitive inhibitor. You don't need this diagram, so I'm just trying to show you the example or make you clear about competitive inhibition. So this is an enzyme succinate dehydrogenase, let's say, and this is a usual substrate succinate, and this is an inhibitor malonate. So you can see here the structure are almost similar. One is some functional groups are missing, but the structure is similar. So this enzyme can bind with the succinate. If it binds with the succinate, then it is converted to fumarate, which is a product. But if it binds with the malonate, there is no product formation. That is, the enzyme will be inhibited because the active site cannot discriminate between so that the same active is this is a HMG coenzyme, which is a usual inhibitor, lovastatin. Now you can see here the structure is quite different here. It is bigger and it is smaller, but at the certain point the structure is exactly same. You can see here HMG coenzyme and this one structure is almost similar. So despite of binding with HMG coenzyme, this enzyme will bind with the lovastatin competitively, so that the enzyme will be inhibited. So these two diagram exemplifies how competitive inhibition works. Another is uncompetitive inhibitor. Although this inhibition is very rare in the biological system, however there are certain conditions where uncompetitive inhibition also works. So the inhibitor will bind only to the enzyme substrate complex, preventing product formation. This one is enzyme, this one is substrate, and this one is the active site. Substrate is normally binding to its active site, and there comes inhibitor which binds with the allosteric site, that is other than active site in an enzyme. Here the substrate and inhibitor are not structurally related, they are very different from each other, so that there is no any competition by site. But inhibitor binds to another site in the enzyme, whereas substrate binds to the active site due to their structural difference. So when inhibitor binds, the enzyme cannot function properly. Although there is substrate, although it is binding to the substrate, but still there is a presence of inhibitor. And here the inhibition is not reversed by increasing substrate concentration. So even if you increase the concentration of substrate, the enzyme will not function. So because the active site is not occupied by the inhibitor. Inhibitor is binding in somewhere else. So this is an uncompetitive inhibition. Example, glyceraldehyde 3-phosphate dehydrogenase inhibition by arsenate. So the inhibitor still here binds non-covalently. So in some point of time the inhibitor will go out and come in depending upon the concentration of inhibitor. Another is non-competitive inhibition. Again, similar to uncompetitive inhibition, there is no structural resemblance between inhibitor and substrate. However, in this case, the substrate normally binds with the enzyme to form enzyme substrate complex, and inhibitor also binds with the enzyme independently. The binding of substrate and inhibitor is independent. Inhibitor can either bind with the free enzyme or it can bind with the enzyme substrate complex. But in uncompetitive inhibition, the inhibitor only binds with the enzyme substrate complex. But the difference here is the binding of inhibitor and substrate are independent of each other. Inhibitor either can bind with the free enzyme or with the enzyme substrate complex to form EI or ESI. In both cases, the enzyme is inactivated. So this is a non-competitive inhibition because there is no any competition. Example of non-competitive inhibitors like heavy metals inhibiting the enzyme. These heavy metals bind with the specific amino acid in the enzyme inhibiting it. Ethanol inhibition of acid phosphatase, anti-cancer drug inhibiting nucleotide synthesizing enzymes, and alanine inhibition of pyruvate kinase. So these are some of the examples of non-competitive inhibitors. Now another is irreversible inhibition. Now here inhibitor binds enzyme, permanently inactivates them. In the previous section, the bond between enzyme and inhibitor was non-covalent. So it can break depending upon the condition of cell, depending on the ionic concentration, pH, or different factors that can that can remove the inhibitor from the enzyme because of the non-covalent linkage, which is much weaker. In irreversible inhibition, there is a covalent interaction between enzyme and inhibitor so that the enzyme is permanently inactivated. Usually these inhibitors are called poisons or toxins. An inhibitor cannot be removed. It is always permanently present in the enzyme, permanently remain attached with the enzyme. The binding, however, can be competitive or non-competitive. That is, the inhibitor can irreversibly bind either with the active site or on the non-active site. That is the active site or allosteric site. Active site is where the substrate binds, and allosteric site is the site other than the active site where different molecules will bind. So they can react with specific R group of the amino acid in enzyme, or themselves have reactive group or activated by metabolism. So there are three mechanisms. These inhibitors can directly react with the amino acid, deactivating enzyme, or they may themselves be reactive. They may contain reactive group which can also affect the enzyme activity, or sometimes they needs to be activated by metabolism. So in their original form they are inactive, but when they are metabolized by the enzyme which they are inhibiting, then they will convert themselves into inhibitor. That is called metabolic activation. Now you can see here examples of irreversible inhibitors. For example, enzyme acetylcholinesterase, it is irreversibly inhibited by diisopropyl fluorophosphate DFP, which is a NORP gas. Or malathion, it is a pesticide which is normally used in crop field causing paralysis. So they will inhibit the acetylcholinesterase, which is required for muscle contraction and other neurotransmission. So in the presence of these inhibitors, the neurotransmission stops and there is a paralysis. And that paralysis is permanent. It cannot be reversed back. Another is cytochrome oxidase. It is an end enzyme of electron transport chain and it is poisoned by cyanide. And the binding of this cyanide is permanent so that the whole respiration will stop. I mean, immediately person dies. Another is glyceraldehyde 3-phosphate dehydrogenase. It's an enzyme of glycolysis, and this is inhibited by iodoacetate. Similarly, aldehyde dehydrogenase is inhibited by disulfiram. Disulfiram is used to treat alcoholism. Normally, aldehyde dehydrogenase will metabolize alcohol to produce acetaldehyde and other aldehydes. So in the presence of disulfiram, this enzyme is inhibited, so that there is accumulation of other substrate which will avert the person from taking excess alcohol. Another is dimercaprol, which is a chelating agent which is used in the treatment against heavy metal poisoning. This will competitively binds to the sulfhydryl group of enzyme, preventing heavy metal binding to them. Another is cyclooxygenase. Which is inhibited by aspirin, which is an anti-inflammatory and analgesic drug. So it is also competitively inhibits cyclooxygenase, and thus the synthesis of inflammatory or pain-inducing prostaglandins are not synthesized. Another is penicillin, which is antimicrobial. It will inhibit the bacterial transpeptidase. Another is fluoride. It is an antiglycolytic, and that will inhibit the enolase, which is an enzyme of glycolysis. So there are just few examples of different drugs or molecules which irreversibly inhibit the enzyme. Now another is suicide inhibition or mechanism-based inhibition. This is a special case of irreversible inhibition. A special case of competitive irreversible inhibition. It means that the inhibitor binds with the active site of an enzyme, and that binding is irreversible. Normally inhibitor and the substrate are structural analog. We have already studied competitive inhibition, where substrate and inhibitors are structural analog of each other. The competitive inhibitor is metabolized to a toxic product. That is, these inhibitors require metabolic activation. In its original form, it is non-reactive, but once it is metabolized, it forms a toxic product, and that product irreversibly inactivates the same enzyme which is used for its metabolism. Now, for example, xanthine oxidase. Xanthine is a usual substrate, and the inhibitor is allopurinol, which is a competitive inhibitor of this enzyme. And when allopurinol binds with xanthine oxidase, this allopurinol is metabolized to alloxanthine. Now this alloxanthine is now another is thymidylate synthase. The usual substrate is deoxyuridine monophosphate, and the competitive inhibitor is 5-fluorouracil. And there is a formation of fluorodeoxyuridine monophosphate formed. So there is no synthesis of thymidine. So it means that abnormal bases are incorporated during the synthesis of nucleic acid, which will disorganize the structure of nucleic acid. That is effect in the nucleic acid formation, mainly DNA. Another is arachidonic cyclooxygenase again. The arachidonic acid is the usual substrate, and the inhibitor is aspirin. So when aspirin binds with the cyclooxygenase, the enzyme gets acetylated, and that acetylated blocks the entry of arachidonic acid inside the active site. Another is HIV reverse transcriptase. So there is an inhibitor, zidoburin, which is an antiretroviral. So it modified when this zidoburin is incorporated into the DNA molecule, then the structure of DNA will be disorganized. The enzyme reverse transcriptase will be inhibited. So these are cases of suicide inhibition. Now you can see in this diagram. So you don't need this diagram to draw, just to clarify the concept. This is a hypoxanthine, and this is a xanthine, which is formed from hypoxanthine, and there is an enzyme xanthine oxidase. This one is a xanthine oxidase enzyme. So when xanthine binds with the active site of this enzyme, there is a formation of enzyme substrate complex, and the xanthine will be converted to uric acid. This is a normal metabolic process in the presence of normal substrate xanthine. But now there comes allopurinol, which is a competitive inhibitor of xanthine oxidase. Now xanthine oxidase will convert it into alloxanthine, which is also called oxypurinol. Now you can see here this structure, this one portion, which mainly interacts with the active site. The structure is almost similar. So both xanthine and alloxanthine can bind with the active site of enzyme because of the structural resemblance at this portion. When xanthine binds, it is transformed to uric acid, but when alloxanthine binds, it cannot be converted to product. The complex of alloxanthine enzyme will inactivate the enzyme because alloxanthine cannot be converted to any other product. But in case of xanthine enzyme complex, xanthine is a usual substrate, so it is converted to uric acid. So there is a suicide inhibition. The enzyme is simply doing a suicide by binding with the inhibitor. It cannot discriminate whether it is its own substrate or whether it is a toxin or inhibitor. So it binds readily with the inhibitor. But by the end, the allopurinol itself becomes a toxin-like substance and it will permanently inactivate the enzyme. This is a case of reverse suicide inhibition, which is a special case of irreversible inhibition. Non-competitive inhibition. Usually inhibitor is a regulatory molecule in this case. We are not using any drugs, poisons or toxins. We are simply using the regulatory molecules. And there is a reversible non-competitive inhibition. Now this is an enzyme and this one is the active site. As the allostatic implies, there are two sites in an enzyme. One is active site, another is allostatic site. Active site where substrate binds and gets converted to product, and allostatic means non-active site where different inhibitors and regulatory molecule will bind, either activating or inhibiting an enzyme. You can see here the active site contains binding area, catalytic site. There is also availability of cofactors and coenzymes, and it is the site where real substrate binds. And this is the allostatic site of enzyme, which may be near the active site or somewhere in the enzyme. So there can be positive allostatic site or negative allostatic site. If the molecule binds to the positive allostatic site, then that is called positive allostatic effector, which will activate the enzyme. If the molecule binds with the negative allostatic site, then it is called negative allostatic effector, which will normally inhibit the enzyme. So in this way, this allostatic effector, by interacting either with the positive allostatic site or negative allostatic site, they will regulate the enzyme activity. Whenever enzyme has to be inhibited, this one is formed, and this is reversible process. So this is a allostatic inhibition, and examples are, these are enzymes like pyruvate dehydrogenase. The allostatic activator is ADP calcium, which will activate the enzyme, and allostatic inhibitors are the ATP acetyl coenzyme A NADH. They will bind to the negative allostatic site. Similarly, aspartate transcarbamylase, the allostatic activities is a CTP, cytidine triphosphate, and inhibitor is ATP. Phosphofructokinase, which is an enzyme of glycolysis. Allostatic activator is ATP citrate, whereas the allostatic inhibitor is AMP. Similarly, pyruvate carboxylase, the allostatic activator is ADP, whereas the allostatic inhibitor is acetyl coenzyme A. So you can see here, actually the allostatic inhibition is a way of regulating enzyme activity. Whenever during the metabolic process, with that the whole metabolic process is an enzyme catalyzed reaction, and there are a battery of enzymes. And by regulating the activity of enzyme, the particular metabolic pathway can either be activated or inhibited. So by targeting certain enzymes, these are the molecules which will target the enzymes of each metabolic pathway. So depending upon the need of our body, depending upon the resource available in our cell or in our body, these allosteric activators are present. Sometimes the concentration of these activators exceeds, and sometimes the concentration of these activators becomes higher. So whichever wins will win the race. So if there is higher amount of these activators, the enzyme will be activated, so the pathway will go on. So if the concentration of these molecules is higher, then the enzyme will be inactivated, so that the pathway will slow down or will stop. So, and this is done to regulate the biochemical mechanism of our body. Now this shows the list of drugs that are used against enzymes used for therapeutic purpose. For example, you can see here methotrexate is an anti-cancer agent. It is an inhibitor of dihydrofolate reductase. It is used in chemotherapy to inhibit the DNA synthesis in tumor cells. Similarly, aspirin, it is used to inhibit the synthesis of prostaglandin by inhibiting cyclooxygenase 1 and 2. Sulfonamides, it is antifolate drug, inhibit the folic acid synthesis in microorganisms. So it is an antibacterial agent, antibiotic. Allopurinol, it is used against gout. Gout means it is an inflammatory condition which is due to excessive production of uric acid. So these uric acids gets deposited into the in the joints, leading to severe arthritis or joint pain due to the excessive deposition of uric acid that forms crystals. So in the presence of allopurinol, the uric acid formation is decreased by inhibiting the enzyme xanthine oxidase. So this you have already seen in the diagram how the allopurinol inhibits xanthine oxidase. Another is statins. You also saw about the role of statins, how it inhibits the HMG coenzyme reductase competitively, and it is used to lower the blood cholesterol level. And another is dicumarol. So you have already seen about the dicumarol. It is an anticoagulant agent. It prevents the activation of vitamin K. So in the absence of active vitamin K, blood clotting factors are not activated, so that the usual process of blood clotting slows down. So it does not allow the blood to clot easily, dicumarol. So usually it is especially given to those patients where there is higher, who are under the risk of thrombosis. So this will prevent the formation of unnecessary blood clot by inhibiting the process of blood coagulation. So this is all about the enzyme inhibition. So I hope you have understood about enzyme inhibition. So if you have any confusion, you can ask. Keisa sonu bane yasamma. Anyone? Anyone have any confusion?